Good morning students. Today we will be learning about immunology, a very important and a very interesting topic that we are going to discuss here. This is a PG, post graduation or a graduation level study, and for those who are willing to acquire knowledge on this topic. So, as you can see, we are going to start today the topic of immunology. So, immunology or immunity, what does that mean? The term immunity has come from the word immunis. It's a Latin word which means exempt. This is a Latin word. So, what does this exempt means? This exempt means exemption of the microbes or pathogens from the body. Okay, so exempt means the exemption of the microbes or pathogens from the body. So now immunology. So now immunology is the study of immunity and immune system. Edward Jenner is known as the father of immunology. Okay, so why Edward Jenner is known as the father of immunology? He was the first to introduce the vaccine system. So how did he do that? He first observed that there were milkmaids working in a dairy, and the milkmaids were prone to a small or a minute disease called as cowpox that often happened to the cows. They were prone to this cowpox and already had this. Again, in later days, when a severe form of this pox known as smallpox came into existence, all were affected by this. All populations were affected by this, but these milkmaids who were already infected by this cowpox were not affected by smallpox. These population, the milkmaid populations were not affected by this cowpox. How did this become possible? So this was possible only due to immune system of the milkmaids. The milkmaids had developed immunity against the cowpox, which was similar to the severe kind of disease that is the smallpox, and hence they were immune to that disease. So this was the whole story, how Edward Jenner introduced the vaccine. Next comes the immune system and its types. The immune system is composed of cells, then organs, then proteins, then tissues. These are the compositions of the immune system. So, which types of cells the immune systems are composed of? They are one is your granulocytes, another one is your agranulocytes. Okay, so these are coming under the WBCs. Okay, simply known as the leukocytes. Okay, granulocytes, under granulocytes we have eosinophil, basophil and neutrophil. And under agranulocytes we have lymphocytes, monocytes and lymphocytes. Lymphocytes are again of two types. They are B lymphocytes and T lymphocytes. Okay, so these are the cells that are coming under the immune system. Again, many others different cells come under immune system, but we are going to study them in the next classes. So we are also going to study about these cells in details in the next classes. So first, what are granulocytes? Granulocytes have granules in their cytoplasm, hence called as granulocytes. And agranulocytes do not have any kind of granules. Simply. Okay. Then organs. What are the organs of immune system? Suppose take an example, that is spleen. It is an organ of immune system which acts for storage of immune cells. Lymph nodes. Okay. Lymph nodes are again another organ of immune system. Thymus is another organ of immune system. Okay. Proteins. What are the proteins in the immune system? You can take example as chemokines that act upon different types of cells of immune system and are produced by the different types of cells of the immune system, and cytokines. These chemokines and cytokines, these are different types of proteins of the immune system. Then tissues, if we come across this lymphatic vessels, okay, these are the tissues that help in the maintenance of the immune system. Okay, so this is the all components of the immune system. Again, the immune system is of 3 levels. What are those? If we can call it 3 levels, we can call it the 3 levels of defense of immune system. So what are the 3 levels of defense of the immune system? First is your physical barrier. First is your physical barrier. Second is your innate immunity. Third is your acquired immunity. We will be learning about all of them. First we will learn about innate immunity. I had told that there are 3 levels of immunity. First is your physical barrier, second is your innate immunity, third is your acquired immunity. But the physical barrier actually comes under the innate immunity. The innate immunity involves the physical barrier. So what are the physical barrier? The skin itself acts as a physical barrier. This prevents the external bacteria, the external environment to be separated from the internal body parts and internal body system. Okay, so the skin first is the primary thing that protects our body. And we can also tell that the skin, which has ectoderm, or we can tell as the ectodermal layer, which separates our body from the external environment is the physical barrier. It is the first line of defense. It is the first line of defense. Okay. Then comes the physiological barrier. In physiological barrier, there comes into existence the HCl in the stomach. First of all, lysozymes in the saliva, then HCl in the stomach, then comes the sebum secreted by the skin. Okay, the respiratory and the reproductive tract. These contains mucus which traps the bacteria. So this is the second line of defense. Okay, this is the first line of defense, this is the second line of defense. So then, in a broad term, we can call innate immunity or the native immunity as native immunity. Why? We can call innate immunity as native immunity because it is present from our birth. The acquired immunity or the adaptive immunity comes there into existence after our birth, when we start developing antibodies, T cells, B cells against the antigens. We start developing all these T cells and B cells, and which comes under the adaptive immunity. This comes into existence after our birth. Okay, and after a certain time after the birth. Okay, so native immunity is present from our birth and plays very important role in the immune system. Native immunity is a non-specific type of immunity. It is non-specific type of immunity. This means it is not specific towards any kind of bacteria or pathogen. It acts upon when a pathogen or a toxin enters our body indistinctively. This means that it is non-specific. Any bacteria, any pathogen that comes inside our body which is foreign to the body, it acts upon it without any discrimination. It may be good, it may be bad. Means the bacteria may not harm or may be harmful, but it clears out the bacteria. This is the primary role of the native immunity or the innate immunity. So it immediately comes into action. It is first acting, first or fast acting, first acting and also fast acting. It comes into play the immediate moment after infection. Only just after the infection it comes into play. Okay, so what are the examples of this innate immunity or native immunity? So examples we can take as the PRRs, pattern recognition receptors. Okay, so we can also tell that NK cells, they also come under innate immunity. Natural killer cells, these are natural killer cells. Then the chemical barriers that I have told, these are the chemical barriers or the physiological barrier, then the physical barriers, these all come under the innate immune system. Okay, then It also includes a series of pre-existing serum proteins. It also includes a series of pre-existing serum proteins which are referred to as complement system. These complement system bind to the PAMPs, pathogen associated molecular patterns, PAMPs, pathogen associated molecular patterns. So these PAMPs, they get attached to the complement system and initiate a cascade of events which are destructive in nature. These PAMPs attach to the complement system and initiate a cascade of destructive events. Okay, these destructive events result in the destruction of the foreign particle or the pathogen. Now come to what is an antigen and what is an immunogen. Any foreign particle in our body is called as an antigen. But what is an immunogen? Immunogen is any antigen that excites immune response. Okay, so immunogen is any antigen that excites immune response. Every antigen does not excite immune response. Okay, every antigen. There are many antigens that we counteract with daily, but every antigen do not excite the immune response. Some antigens excite the immune response. So these antigens that excite the immune response are called as immunogens. Okay. Next is your adaptive immunity. Or simply it can be called as acquired immunity. So what is this adaptive or acquired immunity? So this adaptive or acquired immunity is the third line of defense. This is third line of defense. It is much more specific, very specific. Okay, so minute molecular differences also is recognizable by the adaptive or the acquired immunity. If a pathogen, simply a COVID strain, if you take different COVID-19 strains, okay, coronavirus, we had 2 or 3 strains now in the market. We are having 3 strains. Okay, so the 3 strains are having different kind of molecular patterns. They are structurally different. Okay, so this immune system elicited is also different in all the 3 kinds of that virus. So this is very, very specific. It is particular for the particular antigen. Okay. So it relies upon B and T cells for the immunity. Okay. So it mostly acts, this immune system mostly acts 5 to 6 days after antigen counter. So this is a very long time. Why this takes a long time? Because it takes time for the activation of the T and B cells. The activation of T and B cells for the particular antigen or pathogen takes a lot of time. So it is a long time. It takes about 5 to 6 days. So when we counter a viral infection, it is always said that it is going to take minimum of 5 to 6 days, okay, to get cured. Because when we have a viral disease, we do not take any kind of antiviral medicines. We only take azithromycin, which is an antibacterial. When we are encountered by a virus, our immune system is already down. So if any bacterial infection takes place, it might get severe. So to stop the bacterial interaction with our body, we are taking the antibiotics. So these antibiotics are antibacterial in nature. These are not antiviral. Okay. So after also we take these antibiotics, the disease is going to stay for 5 to 6 days. The only reason for this is that it is cured only by our immune system, and the immune system takes 5 to 6 days for the action, for its action. Okay, so the activation of the B cell or the T cell, the T cell activation takes place. It results in the formation of cytotoxic T cells that counter with the antigen and destroy it. Again, the B cell activation takes place. It produces what? Antibodies against that particular pathogen or the antigen. Then antibody antigen interaction takes place and the antigen gets destroyed or the pathogen gets destroyed. So this process is your time taking process. Okay, this activation of the B cells and the T cells. Then again, so we can give the example here as B cells, T cells. Okay, B cells produce antibodies. T cells are the cytotoxic T cells, TC. Okay, activation which kills the engulfs bacteria or the pathogen. Okay, so this is all about your adaptive immunity or the acquired immunity. Now let us learn about immunity in brief. Immunity, if we tell that we have two types of immune system. First is your innate immunity, another one is your adaptive immunity. In your innate immune system, we have physical barrier, we have physiological barrier, we have cellular barrier. Cellular barrier, your NK cells, these are the cellular barriers. Your macrophages comes under also innate immunity. Okay, cellular barrier, your NK cells, macrophages, dendritic cells, all these comes under innate immunity. Okay, so next your cytokine barrier. Cytokine or chemokine barrier. Okay, so these three comes under these four: physical, physiological, cellular, and the cytokine barrier comes under the innate immunity. Then comes your adaptive immunity. Adaptive immunity is also of two types. One is your active immunity, another one is your passive immunity. Okay, active immunity is again of two types. One is your natural, another one is your artificial. This is again of two types. One is your natural, another one is your artificial. Okay, so what is active natural immunity? Your active natural immunity is your immunity that is developed on encounter upon pathogens directly. Okay, so this is called as natural active immunity, immunity that develops on encounter of the pathogens directly. Okay, suppose you have encountered smallpox once, you will never encounter smallpox again. Okay, this is because you have natural immunity developed against this smallpox virus. So you have active natural immunity against this virus, smallpox. Again, what is artificial active immunity? Artificial active immunities are the antibodies produced against a pathogen as a result of vaccination. So we often deal with this vaccination as passive immunity. No, it's not. The vaccination is actual a part of active immunity, but it's artificial active immunity. Vaccination, remember this, vaccination is a part of active immunity, but it is artificial active immunity. It is the immune response or the immunity against a particular pathogen is developed artificially by vaccination. Okay, so again we come to this passive immunity. So what is the passive immunity? So passive immunity, which is again of two types, artificial and natural. So what is your natural passive immunity? Your natural passive immunity is the antibodies passed on to the fetus by the mother's milk. Okay, what is passive artificial immunity? The anti-venom, suppose we take example anti-venom that we take. Okay, or antibodies, it may be monoclonal antibodies or any kind of antibodies that are extracted from another host and is given to our body, okay, against to counter infection, is called as artificial immunity. Okay, suppose simply if we take a case of snake bite, take a case of snake bite, what happens when a snake bites? Toxin of the snake enters our body. So against it there must be antibodies so that to neutralize those toxins. For the neutralization of the toxins that are produced, that is injected to our body, we need antibodies, but we cannot generate it spontaneously because it takes 5 to 6 days. But within a minute or within an hour or two, the person will be dead. So for that reason, we have to inject antibodies that are developed previously, okay, against that particular toxin, and it is injected, that anti-venom or the antibodies that are injected directly to the person that is infected or that is snake bitten, so that it can be saved. Okay, so likewise the snake bite is an example of artificial passive immunity. Okay, so now you understood the whole concept of adaptive immunity and different types of adaptive immunity. Adaptive, passive adaptive immunity, active adaptive immunity, then passive adaptive of again two types, natural and artificial. Active adaptive immunity is again of two types, natural and artificial. And the whole misnomer of this vaccination being described as passive immunity, which is not actually passive immunity, it is a part of active immunity, but it is artificial active immunity. Okay, Now let us come to the types of immunity based upon the immune response. Immune response is generally of two types. One immune response is your humoral, another one is your cell mediated. Immune response, humoral immune response. So this is also known as in short form CMI. Okay, what is humoral immune response? Humoral immune response simply refers to the immune response which is raised due to the macromolecules present in the extracellular fluid, ECF. The macromolecules that are present in the extracellular fluids. So we can refer here to the antibodies. Okay. So what happens here exactly? What happens here is intact antigens counters with the B cell. Then the B cell then give rise to, one is your plasma cell, another is your memory B cell. Okay, these plasma cells secrete antibody. Okay, the memory B cells, these are stored in the form of memory. This elicits immune response in second infection. Okay, for the first time when an infection takes place, all these events takes place and it takes about 5 to 6 days. Okay, so but in case of second infection, the elicitation of immune response is much quicker than that of the first infection. Why it is much quicker? Because these memory cells are stored, okay, which when triggers, the second infection gets elicited very quickly, okay, and immune response starts acting upon it. Then in case of cell mediated immune system, CMI, what happens? TC or cytotoxic T cells comes to the play. TC is again of two types. One is your memory T cells, another one is your cytotoxic active T cells. These active cytotoxic T cells help in the killing of pathogen. Hence it is called as cytotoxic, toxic to the cell or pathogen. Okay. So again, what happens? These antigens, intact antigens, first interact with B cells. Again, antigens that are engulfed by macrophages, which are the innate part of the what? Innate immune system comes into play. These antigens, then macrophages displays these antigens on their surfaces to the T helper cells, TH cells. So this TH cells, then what it does? It indirectly elicits the B cells. It indirectly elicits the B cells for its proliferation into plasma cells. Is that clear? Again, these T helper cells get converted into Th memory cells, and some T helper cells get again promote the proliferation of the T helper cells itself. Some T helper cells counters with cytotoxic T cells and help in the generation of cytotoxic T cells also. Okay, so this is the whole process how an immune response is generated. So from here we can now conclude that there is a close relationship between humoral immunity and cell mediated immunity. And what is the mediator between them? T helper cells is the mediator between these two types of immunity. Again, we can also say that innate immunity also has a very important role in the action of adaptive immunity. How? Because the macrophages and the dendritic cells helps in the elicitation of adaptive immunity. Hence, we can also say that there is a close relationship between innate immunity and adaptive immunity. Okay. Next comes into the play, that is your PAMPs. PAMPs are nothing but pathogen associated molecular patterns. So let us understand first what are PAMPs. PAMPs are pathogen associated molecular patterns. These are simply the molecules that are either released by the pathogens or the molecules that constitute the pathogens. So when a pathogen is encountered by a macrophage or a dendritic cell or any kind of phagocytic cells, it captures and engulfs the pathogen, and using the lysozymes bombardment it kills it. But the constituents of that cell or the pathogen remains inside the macrophages. So these macrophages or the dendritic cells or any kind of phagocytic cell, what it does, it represents the pathogen using its MHC. MHC is a class of protein, we will be studying it in the next class. So these PAMPs, what they do, they trigger immune response, innate immune system. Okay, so immediate attack on such molecules or pathogens takes place by macrophages or DCs. So DCs are the dendritic cells. Okay, so using enzymes, proteases, lipases and other enzymes. Okay, so this degradation takes place. Lysozymes. Okay, so this degradation takes place. Okay, so these after this degradation of the pathogen, suppose this is a macrophage, it has engulfed, this is either a PAMP, okay, or any pathogen. Suppose this is a pathogen, this is engulfed by the, suppose this is a DC, dendritic cell, it has engulfed this pathogen. Then what it does, okay, dendritic cell, what it does, suppose it collects the PAMP or any DC, it breaks it down and a small molecule is displayed as an antigen to either B cell or T cell for the elicitation of immune response. Okay, this is the DC, dendritic cell. Okay, so this is the process how PAMs are working. Again, some other things are caused by PAMs. PAMs, when interacts with the DC, promotes DC to release cytokines. Not only DCs, we can call them as any macrophage. Okay, so this promotes the macrophage to produce cytokines against these PAMs. The cytokines are messengers that promote killing of the pathogen by inviting other macrophages. Okay, so cytokines, what it does, it sends a chemical message to other macrophages. Suppose this is a border, there is an attack on the border, some soldiers have turned into this side, so one has encountered, here is one Indian border security force, and this particular person has encountered that some from the across the border have entered into the territory. So what will this particular person do? This will try to alarm the others. So this cytokine processing and release of cytokines is the same process of alarming. What does this? Does this simply alarm the other macrophages that some infection has occurred, and hence other macrophages come to the infected site and start killing the pathogens. Okay, now we'll talk about overview of immune system. Or immune response. How an immune response takes place. For an immune response to take place, first thing is your encounter of pathogen, or simply pathogen encounter. The pathogen is first encountered by the innate immune system. Then macrophages, which are the part of innate immune system, engulf the encountered pathogen. This then chops and destroys the pathogen, okay, using lysozymes and many other enzymatic proteins. So again, what these macrophages do, these macrophages or DCs, okay, there are two different terms. Let me tell you here, there are two different terms. The macrophages include two different types of cells. One is your neutrophils, another is your monocytes. These two cells are phagocytic in nature. These help in the phagocytosis of different pathogens. So, macrophages are very tissue specific. This means that they do not leave a tissue. They remain in a particular tissue. Suppose this is a lung. Inside the lungs, you will see lung macrophages. They will not be found, these lung macrophages, they will not be found anywhere else outside the lungs. They will be found only inside the lungs. They are tissue specific in nature. Suppose this liver, it has liver macrophages. They cannot be found anywhere else rather than the liver. So these are very tissue specific in nature. But some are there, some types of cells like DCs I have told about, the dendritic cells. Dendritic cells, we had also learned about dendritic in the neuron chapter, which simply means that its structure is changeable. The cell has such a structure that is changeable shape. This cell can change its shape. So hence it has high mobility. Or you can tell it is a mobile cell. It can move from this tissue to other tissue and so on. It can move from lungs to the thymus. It can move from liver to the thymus and from anywhere of the body to anywhere. Okay? Through the lymphatic system. So this DC, which is very mobile in nature, helps in the transmission of pathogens that it holds with its receptor known as MHC and reaches the lymph nodes or the thymus for the detection of the pathogen by the T cells or B cells. This is simple as that. So DC is known as professional APC. DC is known as professional APC. What is APC? APC simply means antigen presenting cells. What are antigen presenting cells? Simply this means that the cells that present antigen to the T cells or B cells for production of either antibodies or cytotoxic T cells against that particular antigen or pathogen. So these are known as APCs. Okay, so these present the antigen to the B cell or T cell to make the proper directive defense against that particular pathogen. Okay, so first of all, this was your role of antigen presenting cell, and the DC is known as your professional APCs. So after this, macrophages engulf, there are some residual particles. These are residual particles. These are the PAMs. These are then carried by DC. This is a DC to what? This is an APC, antigen presenting cell. Okay, by DC to T cells. Okay, or B cells directly. Okay, the pathogen also interacts directly. These are residual particles. The pathogen also interact directly with the B cells. The B cells then produce plasma cells, which then secrete antibodies that neutralize this pathogen by destroying it. The T cell, what it does, as I have told earlier, this produces again two types of cells. As I have told you earlier, if interaction of the APC takes place, APC plus cytotoxic T cell, then cytotoxic T cells will be produced. Okay, APC plus T helper cells, then T helper cells will be produced. Okay, so then the cytotoxic T cells and the T helper cells, they increase their number. This also increases its number by clonal expansion. This increases its number by clonal expansion. Simply, it increases its number, the cytotoxic T cells and the helper T cells. This increases its number. Then, simply what it does, the helper T cell then proliferates, help in the B cell proliferation, and the cytotoxic T cells directly act upon the pathogen and kills it, or else the T helpers, which I have already told, it triggers itself for its promotion, and it also triggers the cytotoxic T cells. So this is how a whole immune system works. Okay, so for this class, this much. In the next class, we will be studying more about immunology. Thank you.